Today's programme for the course Biology, Form and Function is called The Mitochondrion and is an account of the current scientific ideas concerning the sources of energy and the way this energy is supplied to the cell.
Peter Mitchell, a biochemist working almost alone in his private laboratory here in southwest England, came up with a radical new theory. His ideas have dramatically changed biologists' views on how the mitochondrion carries out the process called oxidative phosphorylation. This program explains his ideas and their experimental basis.
Today I would like to get under the skin of a mitochondrion, so to speak, to show you how oxidative phosphorylation works. Let's start by having a look at the mitochondrion. In this electron micrograph of a thin-sectioned mitochondrion, you can clearly see that the organelle consists of two distinct membranes, a smooth outer membrane and a highly infolded inner membrane. In addition, there are two spaces. First, the space between the two membranes, which is sometimes hard to see, and the matrix, the space surrounded by the inner membrane. In order to give you a clearer view in 3 dimensions of what the mitochondrion looks like, our artists have prepared a cutaway model of a mitochondrion. Here it is. Oh my God, that's really quite frightening, really. It shows you the outer membrane, the smooth envelope. Second, the space between the membrane. In the middle, you have the matrix space. And most prominent of all, of course, is the highly folded inner membrane. All the reactions of oxidative phosphorylation are only associated with the inner membrane. You really could call the inner membrane the heart of the mitochondrial machinery. However, the matrix space is also involved, although in a somewhat more peripheral way. It contains the enzyme pathways which generate NADH2 and succinate, which, as we'll discuss in a moment, are the major fuels for the oxidation system in mitochondria. Well, how does oxidative phosphorylation work? As the name already tells you, it consists of 2 parts. First, a series of oxidation reactions, and second, a series of phosphorylation reactions. In the oxidation reaction, electrons are split off from reduced substrates and transferred to molecular oxygen. As I'll show you in a minute, this transfer is accomplished by a linear series of redox components. The net effect is the production of a divalent oxygen anion from molecular oxygen. This divalent oxygen anion can then combine with protons that are always around in aqueous solution to form a molecule of water. This sequence of reactions releases energy. The second part is an energy-requiring phosphorylation reaction in which ADP is condensed with inorganic phosphate to form ATP, the major energy currency of living cells. These 2 energy-linked reactions together constitute what we call oxidative phosphorylation. Let me now tell you how the oxidation part of oxidative phosphorylation works. This is relatively easy since this part of the reaction is fairly well known. Most types of mitochondria will only use 2 substrates as major fuel for the oxidation chain, NADH2 and succinate. The electrons are extracted from these substrates by specific dehydrogenases, which work with flavin coenzymes as prosthetic groups, FMN and FAD. These coenzymes become reduced in the process and then transfer the electrons further on to so-called non-heme iron proteins. These are brown proteins which contain iron. The iron is not complexed within a heme nucleus, but bound directly to sulfur-containing groups on the protein itself. These types of proteins are also important in other parts of the respiratory chain. Now the two branches of the chain merge. They both donate electrons to a lipid-soluble quinone called ubiquinone. This quinone is reduced to the hydroquinone. Now the electrons go through a linear series of three red cytochromes: cytochrome B, cytochrome C1, and cytochrome C. Finally, cytochrome C is oxidized by a green cytochrome called cytochrome AA3 with molecular oxygen as the final electron acceptor. For this reason, cytochrome AA3 is also called cytochrome oxidase. Cytochrome oxidase is one of the most complicated enzymes known. It contains not only two spectrally distinct heme groups, but also two copper atoms. Together, these respiratory carriers are called the respiratory chain or the mitochondrial electron transfer chain. If we could look at the mitochondrial inner membrane at a magnification of several millions, which we unfortunately cannot do yet, we would see something like this here. You can see that the membrane continuum is made up of many small phospholipid molecules which are all in rapid motion. You can also see that our studio is not yet fully automated. In this somewhat stormy sea of lipid, there float the proteins. For instance, here you can see two spheres, each of which conforms to one of the knobs seen in negatively stained mitochondria. Each of these spheres is actually a molecule of an enzyme involved in ATP synthesis. Each sphere is in turn attached to a stalk which holds this knob to the membrane continuum. The stalk goes all the way across the membrane to the other side, the outside. You can see it penetrating here. These green units are each one molecule of cytochrome oxidase, the final enzyme in the respiratory chain. You can see that this enzyme too goes all the way across the membrane from the outside to the inside. Attached to cytochrome oxidase, going backwards in the respiratory chain towards the substrate side, we have the red cytochrome, cytochrome C, which is actually attached to the outside of the mitochondrial inner membrane, somewhat like this here. In a similar fashion, the next member in the respiratory chain going towards the substrate is the red cytochrome, cytochrome C1. It is also attached to the mitochondrial membrane close to the outer face, somewhat like this. Note that the carriers are not arranged randomly in the membrane. Going still further, we come to cytochrome B, another red cytochrome. Here we don't know the location too well, but it seems that cytochrome B exists in two similar molecular forms, which together penetrate the membrane from the outside all the way back to the inside. Now look at this. What do you see? You see actually a zigzag arrangement. Electrons would go from the inside via cytochrome B to the outside, then stay on the outside and go via cytochrome C1, C to cytochrome oxidase, and then back again to the inside of the membrane. This zigzag flow of electrons is coupled to the extrusion of protons from the inner face to the outer face of the membrane. What will the result of this be? Obviously, you will accumulate positive charges on the outside of the membrane, and since we are dealing with protons, we will at the same time get a proton gradient, or if you so wish, a pH gradient, being alkaline inside and acid outside. We will now show you an actual experiment on this point. And for doing this, we will use a setup which consists essentially of a closed reaction vessel, which is fitted with a very sensitive pH measuring device hooked up to a recorder. Inside this vessel, we will place an anaerobic suspension of red liver mitochondria, which have been supplemented with the respiratory substrate succinate, and which has been adjusted to roughly pH 7. Since there is no oxygen available, the mitochondria cannot respire and can therefore not extrude protons. The pH does not change with time. It remains at roughly pH 7, in this particular case pH 7.08. If protons had been extruded, the medium would become acid, which in turn would be reflected by a deflection of the recording pen towards the left. The sensitivity of this setup is such that the full-scale deflection of the recording pen would amount to a change of roughly 0.1 pH units. If we now inject an air-saturated salt solution into the closed vessel, there's an immediate acidification of the medium, indicative of proton ejection from the mitochondria. This slow return of the pen, which you now see, reflects back diffusion of the protons into the mitochondria, since the mitochondrial inner membrane is not completely impermeable to protons. We will now repeat this oxygen pulse, and I want you to watch for two things. First, check whether the experiment repeats in that protons are being ejected. And secondly, you should watch what happens to the back diffusion of protons if we add an uncoupler before all the protons have had a chance to diffuse back. Now we add another oxygen pulse, and to our delight the experiment repeats, which is always nice in science. Now we add uncoupler. You can see that upon addition of uncoupler, there's a rapid back diffusion of protons. This was one of the key experiments which helped to make researchers accept Peter Mitchell's chemiosmotic hypothesis. Of the several key points of his hypothesis, the following are most pertinent to our short discussion today. First, electron transport in the mitochondrial inner membrane is coupled to the extrusion of protons from the mitochondrion. How does electron transport drive this proton extrusion? We don't really understand this phenomenon in molecular detail, but it's almost certainly related to the fact that the electron transport follows a zigzag pattern through a respiratory chain whose components are asymmetrically arranged within the mitochondrial inner membrane. A second key point of the chemiosmotic hypothesis is that protons, as well as most other ions in the surrounding medium, cannot passively diffuse through the mitochondrial inner membrane. As a consequence, proton extrusion has two main effects. It sets up a pH differential, delta pH, by increasing the hydrogen ion concentration outside. And it also sets up an electric potential, delta psi, by increasing the positive charge on the outside. The combined free energy of the pH gradient and the electrical potential constitute the high energy intermediate of oxidative phosphorylation, the squiggle. The third key point of the chemiosmotic hypothesis is that protons accumulated outside the mitochondrion can flow back through an enzyme which spans the mitochondrial inner membrane and functions as a specific proton pore. This enzyme synthesizes ATP and is called mitochondrial ATPase. You will hear a lot about it later in the program, and in fact, you have already seen this enzyme. It is identical with the 90 angstrom spheres which cover the inside surface of the inner membrane. This enzyme functions like a proton-driven turbine which couples the energy-yielding downhill flow of protons to the energy-requiring synthesis of ATP. The proton pore in the mitochondrial ATPase can be specifically blocked by the antibiotic oligomycin. This provides a logical explanation for the fact that oligomycin still allows the extrusion of protons, but prevents ATP synthesis. A fourth point of Mitchell's hypothesis involves uncouplers. They facilitate the passive diffusion of protons through the mitochondrial inner membrane, and as a consequence, the ion gradient is dissipated so that no ATP can be made. Let me now tell you how the crucial role of mitochondrial ATPase in oxidative phosphorylation was discovered. This important breakthrough was made by Ephraim Reke and his colleagues in the United States in 1960. You'll remember that the submitochondrial particle is covered on the outside by 90 angstrom spheres. Such a submitochondrial particle can couple electron transport to ATP synthesis. However, if the particle is exposed to strong ultrasound, the 90 angstrom spheres are knocked off the membrane, and the resulting particle can no longer make ATP, even though it can still carry out electron transport. If we now add back the spheres to the depleted submitochondrial particles, they can rebind the spheres, and at the same time electron transport is again coupled to ATP synthesis. In other words, the spheres function as a coupling factor of oxidative phosphorylation. Since this was the first coupling factor to be isolated, Reke called it factor 1 or F1. Each of these spheres proved to be an enzyme which, if assayed in the test tube, could split ATP to inorganic phosphate and ADP. It was an ATPase. It was quite clear to Reke and his colleagues, however, that in the intact mitochondria, this enzyme had to work in the opposite direction. It had to make ATP. We only call it an ATPase out of custom, since its ATP splitting function was recognized before its ATP synthesizing function. Each of these spheres is roughly 15 times bigger than a small protein such as lysozyme. Each sphere contains at least 5 distinct polypeptide subunits, some of them being present in more than one copy. There was a serious problem, however, with this solubilized ATPase. Whereas in intact mitochondria and submitochondrial particles, ATP synthesis as well as ATPase activity are both inhibited by oligomycin. The ATPase activity of the solubilized enzyme proved to be insensitive to oligomycin. The answer to this riddle was quite fascinating. It turned out later that when Reke and his colleagues had detached the ATPase sphere from the membrane, they had left part of the functioning ATPase complex of mitochondria still behind in the membrane. And here it is, this unit which penetrates all the way across from the inner to the outer face of the membrane. This unit does not by itself catalyze an ATPase activity, but it is necessary for binding the ATPase sphere to the membrane and rendering its ATPase activity oligomycin sensitive. Each of these membrane-bound units consists of at least 2 and perhaps as many as 5 different hydrophobic proteins, which together form a transmembranous proton pore through which protons can penetrate from the outside to the inside when they make ATP through the ATPase system. One of these proteins binds the oligomycin antibiotic, and this binding blocks the proton pore and thereby oxidative phosphorylation. In order to study the function of the purified ATPase complex in an environment more closely approaching that of mitochondria, Reke and Kagawa reincorporated partially purified ATPase complexes into artificial phospholipid bilayer vesicles. They first mixed solubilized ATPase complexes, which were of course randomly oriented in solution, with phospholipids and detergents, which are shown in this rather poisonous green here. When the detergents were then dialyzed away, the components spontaneously reassembled into spherical closed phospholipid vesicles in which the ATPase complexes were again oriented as in submitochondrial particles. These complexes could now use ATP to pump protons into the vesicles. Could they also make ATP? No, they couldn't, because there was no proton gradient with the proper polarity which could have driven this function. In order to provide the reconstituted ATPase vesicles with such a proton gradient, Recker and his colleague Stokinius did the following fascinating experiment.
One day, Dr. Stokinius from California called me and told me about some experiments which he has been conducting with a bacterium called Halobacter halobium, which lives in high concentration of salt and contains purple patches on its surface as shown in this model. Now he has isolated these purple patches. It's shown that they contain a single protein which was called bacteriorhodopsin, because it shows resemblance to the rhodopsin which is present in our eyes and is required for vision. And he observed that when these bacteria illuminated, that protons move out of the bacterium into the outside. And I suggested to Dr. Stokinius that he come to Cornell University, where I was working, and to bring along some of the bacteriorhodopsin so we can try to incorporate them into our liposomes. And Dr. Stokinius came, and he arrived in the evening, and that evening I took the bottle, the bacteriorhodopsin, which he brought along, which is shown here, a purple pigmented protein, and incorporated into liposome by exactly the same procedure described before, by putting it together with phospholipid and detergent and dialyzing detergent out overnight. Neither Dr. Stokinius nor myself really were very optimistic that the first experiment would be working. So in the morning we took it very easy. We first played a game of tennis, and then, to our surprise, when we performed the first experiment, it worked. Protons were moved when the bacteriorhodopsin vesicles were illuminated. But to our surprise, and rather unexpectedly, the protons did not move out of the vesicles the way they do in intact bacteria, but they moved into the liposomes. And that, of course, made it possible for me to do a rather critical experiment which could answer a very critical question. We could take the liposome vesicles, which contain the oligomyosin-sensitive ATPase, incorporate into it bacteriorhodopsin, and then show that, in fact, when we illuminate it, that the proton would again go into the vesicle, liposome vesicle, be allowed to come out via the oligomyosin-sensitive ATPase and generate ATP. And why this experiment was particularly important is because in bacteriorhodopsin there is no movement of electron, there's only proton pumping, ruling out really the need for electron particle, and this experiment really proved that the chemosmotic hypothesis of a formulation of Mitchell is operative in this model system.
When Peter Mitchell first put forward his ideas in the 1960s, most biologists dismissed them. But in 1978, he was awarded science's highest honor, the Nobel Prize, for his chemiosmotic theory. He had explained one of biology's fundamental processes and given us new insights into the role that the mitochondrion plays in the economy of the cell.